Welcome back, friends. Today we are going to continue with our topic of cytology, and in the previous session we introduced about the cell organelles and the categories of different cell organelles. And in this session we are going to start with the first cell organelle, which is the nucleus, and this is the example of the double membranous organelles. So as I said, that the study of organelles, we divide them into the double membranous, the single membranous, and then finally the non-membranous organelles. So today we'll start with the double membranous organelle, and we'll start with the nucleus. So we'll start the structure of the nucleus, then the functions of the nucleus and the adaptations of the nucleus. So katika kila organelle tutasoma structure, adaptation, then tutaona function za hiyo part au function za hiyo organelle. Lakini pia katika kila organelle tutaona vitu mbalimbali ambavyo vinakuwa associated with that organelle. Kwa mfano tunaweza tunaona vitu kama katika kila organelle tunaweza tunaona vitu mbalimbali yani kwa kila organelle. So what I can tell you that you need to stay tuned. Hapa ndipo ambapo cytology start to be interested topic. Tumeshasoma kuhusu the outer parts of the cell, and now we are digging deeper into the inner parts of the cell. So here they are my contacts, and you can contact me for questions. You can use these numbers in either Telegram, WhatsApp, or you can find me in any way in normal calls or messages for any question or for any part which you have not understood. Now we can start discussing. The nucleus is the center core of eukaryotic cell, so it is the largest structure of the cell organelles. Normally, a cell contains one nucleus. However, in some protozoa, such as Palamasium, there are two nuclei, namely the macronucleus and the micronucleus. The terminology macro simply means it is the bigger one. Macro means bigger one, and the micro means the smaller one. We discussed why we have double nucleus or we have two nucleus in Palamasium when we were discussing about the factors limiting the cell size. And we said that in Palamasium the cell is large, so in order to maintain all activities of the cell, the nucleus must divide into two, so that the macronucleus will tend to control all activities or all metabolic activities of the cell, while the micronucleus will tend to maintain only the reproductive activities and the division of the macronucleus during cell division. So other cells, Palamasium and the Amoeba chaos, they are the two living organisms commonly known to have the two nucleus. But other eukaryotic cells, they have only one nucleus. Prokaryotic cells, they don't have nucleus in such, but they only have the genetic material, which is nucleic acid, which is present in the area within the cell called nucleoid, but not the nucleus in such as in eukaryotic cell. The nucleus is found in eukaryotic cell, but we have exceptions in mature phloem sieve tube element, which is present in the plants. We'll start much about the phloem sieve tube element when we'll be discussing about transportation, but the mature phloem sieve tube element is present in the plants, and the red blood cell in animals. So if you are asked the two exceptions or the two categories of cell where there is no nucleus, in the plants we have the mature phloem sieve tube element, and in the animals we have the red blood cell, which are normally abbreviated as the RBCs. So nucleus is nicknamed as the brain of the cell or the control center of the cell because it controls all activities of the cell. There are some of the questions you will meet with them in the exam, ambayo utakuwa unaulizwa, utakuwa unapewa nickname of the organelle. Ukishapewa nickname of the organelle, then you will be asked to name what is the organelle which is nicknamed in such way. Now, because I want you to understand and to know how to answer different questions, now it's good if I will show you an example of the exam question which is asked in a way that you need to know the organelle and the reason why it is called so. So this was the Form 5 Biology Giants test, which I made it when I was in a water secondary school. So when I was there for temporary teaching in September 2019, I made this paper. I composed this paper for my students. Now we have the question 4 here, ambayo unakuta kuna a nickname of the organelle and then the common name of the organelle, the area of abundant, and the reason. Here we don't have the brain or the cell brain, because cell brain haiwezi kawa na area of abundant. Nucleus is present in all cells. So kuweka nucleus afu kuweka area of abundance unakuwa hauko correct, because nucleus is present in all cells. But the organelles such as post office, the suicide bag, the cell power plant, and the cell store, they are always abundant in some of the cells. So for example, the post office of the cell, the suicide bag, the cell power plant, and the cell store, all of these we'll discuss them in the coming sessions. Na pia you need to know the reason. Kwa mfano unavyosema nucleus is the brain of the cell. Why? The reason is because it controls all activities of the cell. So you may be asking different kinds of questions, but I need you to be aware of all of these kinds of questions which you may be asking. So you have been given the nicknames of some of the cell subunits. Give the common name of each. Name the tissue or organ in mammalian where each subunit is found in abundant, and state why the subunit was given such nickname. Now we can go back to our notes and we can continue the session. I was just showing you an example of the question which you may be asking in this part. So it is very important for everything which I emphasize. It is very important for you to know why do I emphasize that part. Now we can move to start learning about the structure of the nucleus, how the nucleus is, or what is the composition of the nucleus. So the nucleus is typically spherical in shape or oval. It can be spherical or oval, and it is bounded or it is enclosed by a double membrane, which we normally call it the envelope. So it is spherical or oval in shape, and it is bound or it is enveloped or enclosed by the envelope, ambayo tunaita kwa ni nuclear envelope, ambayo inakuwa double membrane. The nuclear poles allow the exchange of materials between the nucleus and cytoplasm. So the envelope is not continuous. Let's say, for example, here is our nucleus. The nuclear envelope is not continuous. So there are some of the poles which allow the exchange of materials. There are some of the poles which allow the exchange of materials. Kwa mfano kama hiyo ni envelope, humu utakuta kuna different poles which allow the movement of materials. Why is this important? It is because some of the materials are synthesized in the nucleus and they are used outside of the nucleus. Kwa mfano tutaenda kudiscuss vitu kama messenger RNA, ambazo tutadiscuss a little molecules like messenger RNA. Kwa mfano the entry and exit of messenger RNA. Messenger RNA anaitwa jina messenger kwa sababu kazi yake ni kucarry message. Ana carry the message normally from the nucleus, and when it carries the message from the nucleus, it send it to the cytoplasm. Normally the messenger RNA inachukuliwa kutoka kwenye nucleus, ambayo messenger RNA inatengenezwa kutoka kwenye DNA. Kwa ikishatengenezwa kutoka kwenye DNA, inachukuliwa pale moja kwa moja. Hii messenger RNA inaenda kupelekwa kwenye ribosomes ambazo ziko kwenye cytoplasm. Messenger RNA ikipelekwa kwenye ribosomes kazi yake zinamconvert messenger RNA kuwa kwenye protein. Tutaenda kudiscuss kwenye detail tutakapokuwa tunasoma genetics, lakini tunachotakiwa kujua hapa ni kwamba in the structure of the nucleus, the double membrane is not continuous. We have the nuclear poles which allow the exchange of materials between the nucleus and the cytoplasm. For example, we have the messenger RNA which tend to exit from the nucleus to the cytoplasm, where the messenger RNA in the cytoplasm is used in the process of protein synthesis. Also, the newly made ribosomes can live through the nuclear poles, and molecules such as proteins and the nucleotide needed for the manufacture of ribosomes and DNA, they enter the cell. Ribosomes zinakamilika utengenezaji wake outside of the nucleus, lakini utengenezaji wake unaanzia kwenye nucleus. Tutaenda kudiscuss kuhusu ribosomes na tutaona ni wapi kwenye nucleus ambako ribosomes zinaanza kutengenezwa. Lakini ribosomes, synthesis of ribosomes starts in the nucleus and then inaenda kukamilishwa kwenye cytoplasm. Also, nucleus itself inavyokuwa inafanya functions kama synthesis of ribosomes, nucleus inakuwa inahitaji enzymes, ambazo hizi enzymes ni proteins. Kwa hiyo hizi enzymes nucleus inazitoa wapi? Hizi enzymes nucleus haitengenezi yenyewe, maana yake inatengeneza messenger RNA. Messenger RNA akifika kwenye cytoplasm anatengeneza proteins, ambazo hizo protein ndio enzymes, because enzymes they are protein in nature. And then hizi proteins zikishatengenezwa kwenye cytoplasm zinarudi tena, so they enter the nucleus again. Their way of passage into the nucleus is through the nuclear pole, cause inaingia tena kwenye nucleus, and in the nucleus they act as the enzymes which are used in the manufacture of ribosomes, or the manufacture of DNA, or even the manufacture of messenger RNA. All of those processes, they require enzymes, which are the proteins produced in the cytoplasm. Kwa tutaenda kudiscuss this process in detail tutakapokuwa tunaenda discuss different topics, na ndio maana tukasema cytology is the basics or is the foundation for you to understand other topics. What we are saying here tutaenda kudiscuss in detail tutakapokuwa tunasoma kuhusu mambo ya genetics, topic ambayo ni ya form 6, lakini you need to have a foundation of cytology. So the outer membrane is continuous with the endoplasmic reticulum, ambayo endoplasmic reticulum is the organelle which we discuss it in detail later in the coming sessions, that is covered with ribosomes for tearing out proteins synthesis. I'm not sure if this is tearing out. I think this is carrying out protein synthesis. So there are mistakes, a lot of mistakes, but we will be correcting them and we'll make sure that they are getting better and better as we are moving further. So this is not tearing, for carrying out. In the protein synthesis process, the organelle which is specifically carrying out the process of protein synthesis, it is the ribosomes. So the ribosomes zinakuwa zimejiattach kwenye endoplasmic reticulum. So tutaenda kudiscuss kwenye endoplasmic reticulum, we have what you call the smooth and the rough endoplasmic reticulum. The rough endoplasmic reticulum is covered with ribosomes, and these ribosomes, they produce proteins. So the ribosomes is for carrying, not tearing, is for carrying. So you consider this and you just insert this word here, it is for carrying out protein synthesis. Kwa hiyo ribosomes kazi yake ni ku carry out protein synthesis. But why are these ribosomes bound to endoplasmic reticulum? Kwa nini hizi ribosomes zisingekuwa free? There are some of the free ribosomes in the cell which we discuss later, but these ribosomes, they are bound to the endoplasmic reticulum because the endoplasmic reticulum function as the organelle which tend to modify the proteins. Kwa hiyo proteins ambazo zimetengenezwa na ribosomes, they enter the endoplasmic reticulum, and by entering the endoplasmic reticulum tend to function as the organelle which tend to modify the proteins made up in the ribosomes. From there, inside the nucleus, there is a matrix called the nucleoplasm or the nuclear sap. The nucleoplasm or the nuclear sap is the jelly-like fluid which contains one or more nucleoli. Together with the chromatin, it is also contains soluble materials of nucleus such as ions, proteins, nucleotides, and enzymes. The nucleoplasm contains chromatin and nucleoli, ambayo nucleoli ni wingi, kama ikiwa moja naita Nucleolus. Unaona, kwa tunakuwa tuna chromatin pamoja na Nucleolus. A chromosome is a very long DNA molecule and the associated histone protein that carry portion of hereditary information. Kwa hiyo what you call the chromatin is made up of the DNA, ambayo DNA imekuwa folded. DNA is like a thread-like molecule. Even the chromosome, they attempted the thread-like molecule, but when they condense during either meiosis or mitosis, they can be observed, because when we strain them, they are observed as in pink color during meiosis or mitosis. Lakini kama hii seli haina condensation ya hizo genetic material, hatuwezi kuziona. They are very smaller molecules in the cell, but they are what which they determine kwamba father amekuwa hivi na mtoto atakuwa hivi because of the genetic materials. So chromosome, they are made up of the very long DNA. Chromosome, they are made up of the very long DNA molecule and the associated histone protein that carry the portion of hereditary material or hereditary information called the genes. You know, when I was studying my O-level, nilikuwa napata shida kwamba what is genes now? They are saying genes. Genes, what is that? I was wondering. Now, okay, yeah, I would like to teach a student in such a way that he will enjoy biology at all. He will enjoy biology na atasema kwamba yes, sijawahi fundishwa biology na mwalimu kama huyu. Now, tunavyosema genes, don't get confused. So I would like to show you what are the genes. Now, this is our cell, and in our cell we have the inside part, which is nucleus. In the nucleus we have the chromosomes, ambazo in magnifying the chromosomes will have this structure ambayo inakwenda centromere, and then tunakuwa tuna telomere. Hizi telomere, centromere ndo part ambayo chromatids zinakuwa zinaungana. So hizi mbili zinakuwa zinaitwa chromatids. Huyu hapa na huyu hapa zinaitwa chromatids. Unaona, zinaitwa the chromatids, zinaitwa chromatids. Kwa hizi zinaitwa chromatids, hapa inaitwa centromere, huku pembeni kuna telomere. Sasa don't get confused with the telomere. Telomere ni piece fulani ambayo katika kila cell division hii telomere huwa inakatika, lakini hivyo vitu achana navyo. Hizo ni concept ambazo tutakuja kuzikuta chuoni, achana nazo kabisa kwa sasa. Don't get confused. What you need to know is that in the cell we have the nucleus, and in the nucleus we have the chromosomes. Chromosomes ni nini sasa? If this chromosome tukiexpand, tutakuja kuona kitu kama hichi. Tukiexpand chromosome, tutakuja kuona kitu kama hichi. Now, in expanding this chromosome, tutaona a DNA molecule, which I hope you have studied it in your O-level biology. So a DNA molecule is a strand of many bases. Many bases. Unaona, kuna base nyingi. Tuna adenine, cytosine, guanine pamoja na thymine. Hizo 4. Lakini hizi base 4 zinakuwa ziko nyingi. They are many in number. Unaona, if they are many in number, finally kitakachokuepo hapa, unaona kitakachokuepo hapa ni kwamba hizi bases they are many in number. Kwa hiyo DNA molecule is very long, composed of many nucleotide and many organic bases. A piece of this DNA, kipande, let's say mimi naanzia hapa hadi hapa, hichi kipande ndio kinaitwa gene. It's called as a gene. Kwa hiyo tunaposema gene, usipate shida kuelewa what is a gene. A gene kitakuwa ni kitu gani? A gene is simply the piece of DNA molecule. So this is known as a gene. A gene is simply a piece of DNA molecule. A piece of DNA molecule. So the piece of DNA molecule is known as the gene. A gene. Kwa hiyo tunaposema gene ni kipande fulani cha DNA molecule ambacho kinakuwa kina carry out information. Kinakuwa kimecarry certain information. There are information which are carried by this piece of DNA. Information hizo zinaweza kuwa kwa mfano labda hair color ya mtoto, either iwe ni black au hair texture, either ziwe kipilipili au ziwe ni nywele ngumu, ziwe ni nywele laini, zinaweza kuwa carried na hichi kipande cha DNA. Kwa sababu hichi kipande cha DNA kimecarry information fulani, hichi kipande tunakiita kama gene. So chromosome, tunaposema chromosome is a very long DNA molecule and the associated histone protein. Kwa DNA molecule with the histone protein, they form what you call as the hereditary information, or they form what you call the gene. The function of DNA is to carry the genes which control different characteristics in living organisms. So in other words, chromosomes are thread-like structure which carry out hereditary information or which carry out genes. So exactly ni kwamba the genes are carried by piece of DNA, or a piece of DNA ndio ambayo inaitwa kuwa ni gene. Lakini kwa sababu DNA with the association pamoja na protein, they form what you call the chromosome. So tunasema chromosome, they carry the genetic material. What do we say about the gene? The gene can be defined as the region of DNA that controls hereditary characteristics. Region of DNA that controls hereditary characteristics. So it is usually correspond to a sequence used in the production of a specific protein or DNA. Unaona? Kipande fulani cha DNA kama hiki hapa kinakuwa kinatumika katika kutengeneza protein fulani. Let's say it is enzyme, or ile protein, let's say it is keratin which make up hairs, yani nywele. Let's say ile protein inaenda kutengeneza skin. Let's say ile protein inaenda kutengeneza the skin color. Let's say the protein it is melanin. All of the protein, their synthesis requires the control of the DNA which is present in the nucleus. Protein zote mwilini, kwa sababu mtu anapokuwa mrefu it is because of the proteins. Kwamba kuna protein ambayo ilikuwa ni hormone. Hii hormone ambayo ni growth hormone imekuwa produced in excess, huyu mtu amekuwa mrefu. Mtu anapokuwa mfupi and dwarf, it is because of the protein. Yani kulikuwa kuna growth hormone iliyotakiwa produced, imekuwa produced in less amount, huyu mtu anakuwa ni dwarf. Unaona? Kwa hiyo all characteristics of the living organisms, they are controlled by DNA, and how it is because of the production of different proteins. Kwa hiyo utakuta kuna enzymes, kuna hormones, kuna chemical messengers. All of these, they are made up of protein, or they are protein in nature. Kwa hiyo tunaposema gene is simply the region of DNA, or the piece of DNA which control hereditary characteristics. Kwa hiyo tunavyoona nywele za mtu ni nyeusi au tunavyomwona mtu ni albino, ni kwa sababu kwenye kipande cha DNA yake kuna kipande ambacho kinasema skin color isiwe black, skin color iwe na rangi fulani. Unaona, ndio tunakuja kumwona mtu ambaye ni albino. So an albino, it's not because yeye ni albino, it's because of the skin color, ambayo kuna piece of DNA which control the characteristics of skin color inamfanya mtu awe albino. So it is usually correspond to a sequence used in the production of specific protein or DNA. A gene carries biological information in a form that must be copied and is transmitted from each cell to its progeny. Progeny means the offspring. Kwa hiyo gene inakuwa na carry genetic information, ambayo nilikwambia inaweza kutengenezwa genetic information ambayo kwa carried by gene, inaweza kutengeneza protein, ambazo hizo protein zina control different characteristics. So the chromatin, tulikuwa tunazungumza chromosomes, unaona, lakini pia tuna kitu tunakiita chromatin. Chromatin is organization of DNA and the basic protein called histone. Okay, very sorry. Here it's not called, it is called. Inaitwa called. So called. Kwa hiyo chromatin ni organization ya DNA pamoja na basic protein ambayo is called histone. So chromatin is composed mainly of coils. Mainly of coils of DNA bound to basic protein called histone. Kwa tunakuwa tuna different coils of DNA ambazo zinakuwa zimebind to histone. Unaona? So DNA is wound around the histone which form bed-like structure called the nucleosome, and these in turn are regularly packed into the chromatin. Unaona? Kwa hiyo unaweza kuona kwamba ni vitu ambavyo kama vinakuchanganya, lakini hivi vitu havichanganyi. What you need to understand hapa ni kwamba inside the nucleus tunakuwa tuna chromatin. Unaona? Ambazo chromatin zinaweza zika condense zikatengeneza chromosomes. Kwa DNA with histone inaenda kutengeneza chromatin. Chromatin zinaweza zika condense kwenda kutengeneza chromosomes. Unaona? So the term chromatin means colored material. Chromatin maana yake colored material. and they refer to the fact that this material is easily strained for viewing with microscope. Ukiwa unaifanyia straining cell with microscope, hii cell itaonesha chromatin zikiwa zina rangi ya purple. Unaona, zina rangi ya purple. Actually this is the diagram, lakini sio diagram ya kuchorwa, ndio maana haionyeshi clear the structure of the nucleus, lakini I think this is the best diagram for studying kuliko zile nyingine zote ambazo niliziona kwa mwalimu Google. For now it is difficult for me to draw my own diagram, but you can visit the biological science book which can show you the very clear diagrams than this. But this is also suitable for our study. Tuna nuclear membrane which is not continuous. Here we have the nuclear poles. Tuna poles hizi hapa. Then the outer membrane of the nucleus, it is continuous with the endoplasmic reticulum, which have the ribosomes. Hizi nyekundu ni ribosomes bound on the endoplasmic reticulum, forming the rough endoplasmic reticulum. So when the messenger RNA, it is made up in the nucleoplasm, it exits out through the nuclear pole and then it binds the ribosomes. After binding the ribosomes, it forms the proteins, and after forming the proteins, the protein tend to move into the rough endoplasmic reticulum. After that, the protein can get modified and then it moves into the channels of the rough endoplasmic reticulum. After that, it moves into the Golgi apparatus. It moves to the Golgi apparatus. Let's continue. There was a problem here, so I was forced to stop the session, and then now let's continue. I hope we ended here. The term chromatin means colored material, and this refers to the fact that these material they tend to strain easier for viewing with microscope. Kwa hiyo tunapokuwa tuna microscope tunataka kuangalia cell, tuki strain sasa tutaenda kuona kuna strain nyingi. Ukifika chuoni utazikuta hematoxin and eosin, which is the commonest strain we are using to strain bacteria. Tuna background strain, Indian ink strain. We have different strains, so I am not here to talk about the strains. Lakini strain tunachanganya cells with a certain chemical to make them visible. So in the chromatins, hizi chromatin huwa zina hold color. Zikihold color manake tunakuwa tunaziona kiurahisi. They appear as deep purple. They appear as deep purple. So by appearing as deep purple, tunakuwa tunaziona kirahisi. So because they are colored material, or in straining they appear as deep purple in color, ndio maana zitakuwa ni chromatin. During nuclear division, chromatin strains more intensely and become more conspicuous because it is condenses into more tightly called threads called the chromosomes. Kwa hiyo always ni kwamba yani chromosome zinaonekana wakati wa nuclear division only. Yani hizi chromatin kama hakuna nuclear division hauwezi kuiona kwa sababu they are very small thread like structure. But during nuclear division, the chromatin they tend to condense. Unaona? Ndio watu tumesema they strain more intensely and become more conspicuous because it condenses into more tightly coiled threads called chromosomes. So during a nuclear division, ndipo ambapo the chromatin they coil up and they become intense, strained. Each nucleus encloses fixed number of chromosomes. Here is each nucleus, not each nucleus. Each nucleus encloses the fixed number of chromosomes, and the number of chromosomes depends on the species. Kwa mfano human being tuna chromosome 46, and because the chromosomes they appear in pairs, tunazita is homogeneous chromosomes. They appear in pairs. Tutaenda ku study in detail kwenye genetics. Chromosomes always appear in pairs. Chromosomes always appear in pairs. So the chromosomes, because they appear in pairs, 46 chromosomes, they make up the 23 pairs of chromosome, in which the 22 pairs they are called the somatic chromosomes, which control other characteristics of the body, while the 2 chromosomes, which is one pair, they act as the sex chromosome. So they control only sexual characteristics, including the formation of the sex organs and the different sexual characteristics, they are controlled by the sex chromosomes. So ndo maana tutaenda kustudy in detail later even in reproduction, ni kwamba human being anapokuwa formed from the first week to the 6th week, huyu human being wanakuwa wanafanana, female and male wanakuwa wanafanana, similar in everything, but the presence of a sex chromosome Y tend to make male to be different to female. Unajua sometimes most of male they don't like to hear kwamba in the first 6 week of development walikuwa similar to female. Most of male they don't like to hear this sentence, but this is the true, and the differentiation of the male structures starts in the 6th week. That means kama kungekuwa kuna lack of Y chromosome, there could be no differentiation of the male structures in the after the 6th week, and then finally tungepata mtoto ambaye ni wa kike. So I don't know how can I explain this, but we'll explain it in detail in reproduction. So we have the 46 chromosomes, 23 of them they are 22 of them they are somatic, only 2 of them they are sexy chromosomes. We discuss about the chromosomes later in genetics again, and we'll discuss more about them later in other topics like reproduction and genetics. So kila species wanakuwa na fixed number of chromosomes. Human being ni different number of chromosomes from pigs, from euglena, from different other organisms. So chromatins are of 2 types. We have the heterochromatin, which remain tightly coiled during interphase and continue to strain intensely, the heterochromatin. So they are largely located towards the peripheral area of the nucleoplasm, towards the peripheral area of the nucleoplasm. The heterochromatin ndio ile ambayo kama ukienda kwenye BS utakuta kwenye structure nucleus kuna baadhi ya parts zimekuwa labeled kama hivi. Unaona, they are always located in the peripheral area of the nucleoplasm. Hizi heterochromatin zenyewe zinabakia intensely strained even during interphase. Remember, during interphase huu ni wakati ambao cell inakuwa high divide. Tutaenda kudiscuss much in cell cycle kwenye topic ya growth, lakini unachotakiwa kujua ni kwamba katika stage nyingi za cell cycle, interphase ni wakati ambao cell inakuwa high divide. Kwa wakati ambao cell inakuwa high divide, hizi genetic material huwa always zinajiachia. Kama unakumbuka nimesema kwamba the chromosomes they strain intensely during cell division, ambazo materials zinakuwa zimebecome close. Wakati ambao cell ya divide, hizi materials always huwa zinafanyaje? Zinajiachia. Materials huwa zinajiachia wakati ambao cell inakuwa high divide. Lakini only the heterochromatin, hii ni category ya chromatin ambayo hata cell ambayo high divide yenyewe huwa inabakia the very intensely strained. Unaona, this is called the heterochromatin. So they are largely located towards the peripheral area of the nucleoplasm. And then you have the euchromatin. Don't read it as euchromatin, that is Kiswahili. So we read it as euchromatin. Euchromatin is a closely coiled chromatin and is largely located, it is loosely coiled. This is tightly coiled. Heterochromatin is tightly coiled, while the euchromatin is loosely coiled chromatin and is largely located towards the center of nucleoplasm. So the euchromatin, they appear as just like lines. In the structure of nucleus. The euchromatin, they appear just like lines kwa sababu zenyewe they are loosely packed. So during interphase hauwezi kuziona euchromatin. This is thought to contain the DNA which is genetically active during interphase. Remember, tumesema interphase ni kipindi ambacho cell inakuwa high divide. Hasa kama cell inakuwa high divide during interphase, hawa euchromatin hawawi coiled sana. Kama hawawi coiled, manake this DNA is genetically active during interphase. Manake hii ndio DNA ambayo inakuwa inafanya kazi ya kuiprepare cell ili iweze kufanya divisions mbalimbali. So this euchromatin contain DNA which is genetically active during interphase. Kwa sababu gani? Kwa sababu heterochromatin inakuwa tightly coiled, maana yake hiyo DNA yake haiwezi ikatumika during interphase period. That is the discussion regarding the structure of the nucleus. Now inside the nucleus we have what you call the nucleolus. Let's discuss a little about the nucleolus before we are moving further. Nucleolus is the round body without a membrane which looks like a large dark spot. It looks like a large dark spot. So I hope you can see the nucleolus here. It looks like a large dark spot. Kwa hiyo ni large round area inside the nucleus which looks like a large dark spot. When you view, not when you received it, hapa ni when you viewed. I am sorry because of the mistakes. Hapa ni when you viewed, with the, or when you viewed by a microscope within the nucleus. So when you viewed with a microscope within the nucleus, the nucleolus tend to look like a dark spot. A nucleus may contain up to 4 nucleoli, but within each species, the number of nucleoli is fixed. It can contain up to 4 nucleoli, but within each species, the number of nucleoli is fixed. The function of nucleolus is to synthesize the ribosomal RNA or the ribosomes. Ambazo ribosomes ni protein producing organelles. The ribosomes ndiko ambako protein synthesis inakuwa inatokea. Mchakato mzima wa kutengeneza protein unakuwa unatokea kwenye nini? Kwenye ribosomes. So it strains intensely because of large amount of DNA and the RNA it contains. Nucleolus contains large amount of DNA and the RNA. That's why it tends to strain intensely. The densely straining core of nucleolus is made up of DNA from one of the several chromosomes. Remember, in the nucleus we have different chromosomes, and these chromosomes, they tend to control different characteristics. As I told you that one pair of the chromosome tend to control the sexual characteristics, but other chromosomes, they control other different characteristics. So the center core of the nucleolus is made up of the DNA from one of the several chromosomes. These contain many copies of genes that code for the RNA needed to make the ribosomes. Kwa hii DNA inakuwa ina uwezo kutengeneza many copies, inatengeneza copies za RNA. Tutaenda kuona kwenye genetics kivipi DNA inaenda kutengeneza RNA, tuta discuss kwenye genetics. But what you need to know is that RNA is always a copy of DNA. RNA is always formed from DNA. So the ribosomal RNA ndio RNA ambayo inakuwa ikiassociate with the protein inaenda kutengeneza nini? Ribosomes. Ambazo ribosomes zinatusaidia katika kutengeneza protein nyingine. So during nuclear division, nucleoli seem to disappear, but this is because the DNA disperses. During nuclear division, there is disappearance of the nucleolus. Au kama kuna nucleoli yani ziko nyingi, they disappear. Now the disappearance is because DNA ina disperse. Kwa hiyo kidisperse hata ukistrain hauwezi kuona tena nucleolus. The reassembly or the reassemblance, yani ile DNA inavyojicollect tena, ikijicollect after division unakuwa unaiona tena nucleolus. So they reassemble after division. Around the center core of nucleolus is a less dense region where ribosomal RNA is beginning to be folded and then combined with protein to make ribosomes. So the ribosomal RNA is beginning to be folded. I'm not sure if this, I will confirm it later, but I think this is beginning to be formed. And then it combined with protein to make the ribosomes. The partially assembled ribosome move out through the nuclear pole into the cytoplasm where assembly is completed. So as I told you that the ribosomes synthesis starts in the nucleus and it is completed in the cytoplasm. We can't synthesize the ribosomes fully in the nucleus. We synthesize it in the nucleus and then we are going to complete it in the cytoplasm. I would like to finalize this discussion by discussing what the functions and the adaptations of the nucleus. Kuhusu functions, kwanza, it contains DNA, which is organized into genes. So the genes, they are controlling all activities of the cell, such as growth, cell division, protein synthesis, and etc. Or sometimes you can say this function as it serves as the information processing and administrative center of the cell. Information processing and administrative center of the cell. Kwa hiyo nucleus ndio admin your cell in nucleus and administrate all activities of the cell. Na ndio maana tukasema the cytoplasm without nucleus is useless, because all of the functions which are taking place in the cytoplasm, they are controlled by DNA or they are controlled by nucleus. Na ukijiuliza kwamba how are these functions controlled? They are controlled by DNA. DNA tend to produce messenger RNA. The messenger RNA tend to take the message from DNA, send it to the cytoplasm, where in the cytoplasm this message carried by the messenger RNA is converted into protein, and this protein can be enzyme, can be hormone, can be neurotransmitter. This protein which is produced, it controls different activities either within the cell or even other parts of the body. Unaona? Kwa unaweza kuona hapo sequence kwamba inaanzia kwenye DNA inatengeneza messenger RNA. Messenger RNA, the name messenger it because it carries the message from the nucleus to the cytoplasm. When this message reaches the cytoplasm, it is converted into the protein, unaona, by the ribosomes, unaona. And this protein might be hormone, or might be enzyme, or might be neurotransmitter, or might be different things. But important thing what you need to understand is that this protein tend to control different activities either within the cell or in different parts of the body. So that's why we are saying the nucleus, which contain DNA, tend to control all activities of the cell, and finally they survive of the whole organism. Number 2, contain the genetic material in the form of chromosome, which control inheritance of characteristics. So the DNA, which is genetic material, tend to control the inheritance of characteristics from the father or from the parents to the offspring. Number 3, it is involved in the production of ribosomes, the ribosomal RNA and the messenger RNA. The ribosomal and the messenger RNA, they are synthesized within the nucleus. Number 4, nuclear division is the basis of cell reproduction and hence, cell replication and hence reproduction. Reproduction yoyote unayojua wewe duniani isingeweza kutokea without nuclear division. Kwa hiyo ni kweli we have the two types of reproduction ambazo tunakuwa tuna asexual and sexual reproduction, lakini even asexual reproduction they depend on cell division or they depend on cell replication. Hata kama ukisema kwamba tunachukua ndizi tunaenda kuipanda ikiwa kama mgomba, still ili ile ndizi iweze kugrow, it must undergo cell replication or it must undergo cell division, unaona. So cell division is the basis of reproduction, and even cell division is the basis of cell growth, is the basis of the organism's growth, because you have mitosis, which always results into the growth of an organism, and you have meiosis, which always results into formation of the reproductive cells. So any category of reproduction which you know in the world requires nuclear division and cell division. Then number 5, all genetic variations are caused by change in the genetic material present in the nucleus. So the genetic variation, they're very important because they contribute to evolution. Now let's move to the adaptations of the nucleus. First, it has the nuclear envelope, which is perforated by nuclear pores, which allow the exchange of materials between the nucleus and the cytoplasm, such as the exit of messenger RNA and the ribosomal subunits, and the entry of ribosomal protein, nucleotides, and other molecules. Now, unachotakiwa kujua kitu kimoja, tunapokuwa tunasoma biology, unapoambiwa utaje adaptation, remember this. When you are required to mention the adaptations, you are required to first start a feature which is present in that organism or in that part of the cell, and then after stating the feature, then utasema ile feature inasaidia nini. Kwa hiyo tunaposema adaptation, manake unataja feature. Baada ya kutaja feature unasema ile feature inasaidia nini? Inasaidia nini ile feature? Kwa mfano tukisema it has a nuclear envelope which is perforated by nuclear pores. Nuclear pores inasaidia nini? Which allow the exchange of materials between the nucleus and the cytoplasm. Hata usipotoa example, it is fine. Lakini sometimes teachers, they require you to give them examples, and giving examples, it is good, because hauwezi kusema nuclear pores while you don't know even what are the materials which exit through the nuclear pore or which enter the nuclear pore. So yeah, it is very easy to memorize the exit of messenger RNA. We have talked a lot about the messenger RNA molecule ambayo inakuwa produced within the cell and then it exit out into the cytoplasm for producing the proteins. Kwa hiyo hata ukikumbuka example moja inakuwa ni vyema. Number 2, the nucleus houses chromosomes containing DNA. Now what is the DNA for? DNA holds hereditary material or hereditary information and instruction for cell growth, development and reproduction. So tunaposema the nucleus houses chromosome containing DNA, now this is not the adaptation. Yani ukiandika adaptation hadi hapa mimi nakuwekea kosa. You need to say what is the function of the DNA or how is that DNA help. So unaweza usiweke full stop, unaweza kwa comma ukasema houses DNA containing chromosome, ukasema which holds hereditary information and instruction for cell growth, development and reproduction. That is fully adaptation. Lakini ukishia hapo kwenye full stop naweka kosa, umekosa. Number 3, presence of DNA associated with protein molecules to form chromosomes responsible for carrying genetic material from parents to offspring. So the DNA holds information, but the chromosome, they are responsible for the transfer of genetic material from parents to offspring. Utanielewa zaidi tutakapokuwa tunaenda kustudy topics kama reproduction, kwa sababu tunapoenda kusoma cell division hatusomi separation of DNA, tunasoma separation of chromatids, ambazo chromatids ni compartment wa nani? Wa chromosome. Kwa hiyo ili DNA iweze kusafiri kutoka kwa parent kwenda kwa offspring, lazima iwe associated with protein histone kwenda form the chromosomes, Number 4, it is covered with the rough endoplasmic reticulum responsible for protein synthesis. It is covered with the rough endoplasmic reticulum. For what? Responsible for protein synthesis. So this is full adaptation. This allows the transfer of materials as well. Hata usipoweka hii mbele, ukishia tu hapo kwenye full stop, this is adaptation inatosha. Number 5, nuclear envelope help to maintain the shape of the nucleus. Number 6, the nucleolus contains nucleolar organizer, the part of chromosome carrying the genes for ribosome synthesis. Unaona? So what is the function of the nucleolar organizer? Is the part of chromosome which carry the genes for ribosome synthesis. So the nucleolus help to synthesize the ribosomes by transcribing and assembling ribosomal RNA subunit. These subunits join together to form ribosomes during protein synthesis. So they join together to form ribosomes during protein synthesis. Here number 6. Number 7, the nucleoplasm supports the nucleus to hold its shape. So it also provides a medium by which materials such as enzymes and nucleotides, ambao nucleotide ni DNA and RNA subunits, tutaenda kwa discuss in detail tutakapokuwa tunasoma genetics, can be transported throughout the nucleus to its various parts. The nucleoplasm contains enzymes which catalyze transcription of DNA into messenger RNA used for protein synthesis. Kwa tutaenda kusoma details zaidi kuhusu nucleoplasm, mambo ya messenger RNA na molecules zingine tutaenda kusoma detail zaidi. This marks the end of our discussion regarding the nucleus. The session was somewhat long, but I hope you have enjoyed the session. This is the way we are discussing about organelles. Now tutaenda kuona mitochondria in the coming session. Tutaenda kuona discussion kuhusu mitochondria. This is also the double membranous organelle ambayo we normally call it the powerhouse of the cell. The session will be enjoyable. Don't miss it. Thank you, and later I wish you nice studies.